Good afternoon. Welcome everyone to Downeast Institute's first climate change seminar of 2022. Thank you for joining us. This seminar is part of our Downeast Discussions series where we discuss and share the different groundbreaking research that Downeast Institute is conducting at our laboratory in Beals and also in the field across the state. And this summer, we're going to be having 6 different seminars, primarily on Tuesdays during the lunch hour of noon to 1. And these seminars are going to be focused on climate change impacts to shellfish. My name is Sarah Randall. I'm the Associate Director of the Downeast Institute. And for those of you that don't know, the Downeast Institute is a nonprofit marine research facility, education center, and shellfish production hatchery in Beals, Maine. And we also serve as the Marine Science Field Station of the University of Maine at Machias. So today's seminar is going to last 40 minutes of presentation, and we're going to end at 1 p.m. So it's very important. This is basically a Zoom meeting. It's not a webinar. So it's very important that people keep themselves muted during the presentation so we can hear Ray present today. At the end of his presentation, we're going to take as many questions as possible, keeping in mind the 1 p.m. end time. You can ask a question by raising your hand on Zoom under reactions or by adding it to the chat. The presentation is being recorded and maybe uploaded to YouTube at a later date. And I would like to thank Hannah Green, who is our Downeast Institute fellow, who is helping with the technology and moderation today. So we're really excited to be able to share today's speakers' work with you today. We're going to hear from Ray Czaja, who is entering his 4th year of PhD study at Stony Brook University. He's one of the inaugural pair of Downeast Institute fellows that we're happy to host here at Downeast Institute this summer. And his research pertains to climate change impacts on the Atlantic surfclam fishery. His approaches range from controlled lab experiments to modeling techniques and aims to assess both individual and population level responses. And Ray does not discriminate when it comes to climate change phenomenon. And he studies ocean warming, ocean acidification, food availability, and wind pattern changes. And so with that, I will hand it over to Ray Czaja for his presentation today about the behavioral and physiological effects of climate change scenarios on Atlantic surfclam larvae.
Great. Thank you for that intro, Sarah. I'm going to assume that everyone can now hear me and see my slides. So as Sarah stated, my name is Ray Czaja. I'm a PhD student in the Marine Animal Disease Lab at the School of Marine and Atmospheric Sciences at Stony Brook University. I'm entering the 4th year of my PhD, and I'm also one of 2 graduate student fellows here at DEI this summer. However, today, I'm going to be talking about our work from last summer involving the behavioral and physiological effects of climate change scenarios on surfclam larvae, and more specifically, how those effects may have dispersal and energy budget implications. Before we get into the science, I want to first start off by thanking and acknowledging the many co-authors involved in this project, including quite a few scientists here at DEI, scientists back at Stony Brook, and even some scientists from other institutes. I also want to thank all the supporting staff here at DEI that helped make the experiments possible. And I want to thank many of my lab members back at Stony Brook who helped me prep for the experiments beforehand and process some samples after the experiments. Now into the science. And first, a brief and familiar background about climate change, more specifically OAW, or ocean acidification and warming. So we have anthropogenic activity releasing carbon dioxide, right? Now, a good portion of that carbon dioxide hangs out in the atmosphere, and it contributes to an enhanced greenhouse effect, allowing more solar radiation to make contact with the surface of the Earth, and in the case of the ocean, leading to ocean warming. However, not all of that CO2 hangs out in the atmosphere. A good portion of it gets directly absorbed by the ocean. And when that happens, it sets off a chain reaction in the carbonate chemistry system known as ocean acidification. And two of the end products of that reaction are reduced pH and a reduced saturation state. And so we have our bivalve shellfish, both adult stages and larval stages. Now these bivalve shellfish, they're ectothermic, so that makes them sensitive to temperature changes. They also have calcium carbonate shells, which makes them sensitive to changes in the carbonate chemistry system. So our bivalve shellfish may be particularly vulnerable to both ocean warming and ocean acidification. And we care about these organisms not only because they provide many ecosystem services, they also support many commercially important fisheries. And the fishery I'm particularly interested in is that of the Atlantic surfclam, Speciula solidissima solidissima. It's a relatively large, broadcast spawning bivalve of 20 centimeters. And they're distributed between Gulf of St. Lawrence all the way down to Cape Hatteras. And here in the Gulf of Maine, you can find them lower in the intertidal, but a little further south in the New York Bight, Georges Bank, Mid-Atlantic Bight, they're primarily a continental shelf species. That's where the fishing happens. And you see them in about 10 to 60 meters of water. You typically hit surf clams before you hit the ocean quahog and sea scallop zone. They are traditionally a capture fishery species, but there certainly are growing aquaculture interests, both here in Maine and further south. And here in the Northeast US, they support a near $30 million fishery where they're often used for fried clam strips and chowder. So we know a lot about population trends for surf clams, and not too surprisingly, most of it's not good. So let's take a look at this figure here. We have landings per unit effort on the y-axis, which you can think of as kind of a proxy for biomass or abundance. And we have time on the x-axis. And we have all the different colors, all the different regions, Southern Virginia, Delmarva, New Jersey, Long Island, Southern New England, Georges Bank, other, which likely includes the Gulf of Maine. And then we have all those regions combined. And so, of course, we see some recent spikes in recent years in some of the more northern regions. But the general trend here is decreasing landings per unit effort through time, almost up to 200 back in the 80s, down to about 50 within recent years. So we have decreasing biomass going on for surf clams, right? And there are plenty of other studies that show decreasing growth through recent decades, decreasing health through recent decades, specifically in the Mid-Atlantic Bight, a little further south. Now, another study that some of my colleagues and I have been working on for quite some time now, looking at recruitment in the New York Bight, has found that recruitment's also been decreasing. So on our y-axis, we have this relative index, unitless. It just shows that a positive value is a high recruitment year, negative value a low recruitment year. And so we have observed recruitment from the late 80s to present day as the black line, and then predicted recruitment as the red line. And so we can see we have a spike here in the late 90s, but the general pattern specifically for predicted recruitment is decreasing through time. The point here is decreasing biomass, decreasing growth, decreasing health, decreasing recruitment. And if we travel a little further south than the New York Bight into the Delmarva region, we can see back in the 80s, we have plenty of surf clams in our inshore region. However, fast forward almost 2 decades later, and surf clams are almost virtually absent from some of our inshore regions. So again, bad things happening for surf clams. So the next question is, okay, well, why is this happening? What's going on here? And perhaps the usual suspect of overfishing comes to mind. However, plenty of surveys and other studies show that overfishing shouldn't be a problem for surf clams. So the next usual suspect up on the list, climate change. Sure enough, there are plenty of studies out there that have looked at climate change responses for surf clams, specifically ocean warming and ocean acidification. And one of the big takeaways from these studies is that adults don't like it warm. And by warm, I mean over 20 degrees Celsius. So Hornstein looked at scope for growth for adult surf clams at 23 Celsius and 19 Celsius. And he found that at 23 degrees Celsius, scope for growth was significantly lower than at 19 Celsius. More specifically, he found that scope for growth was actually negative. So what this means is these animals exposed to these warm waters are using more energy for maintenance than they're taking in. So clearly, bad thing for surfclams. And there are other studies out there at both the population and individual level that show that surfclams decrease growth at these warmer temperatures. And so earlier I talked about my recruitment study I've been working on, and I mentioned that this red line is predicted recruitment, right? What's it predicted from? Well, it turns out observed recruitment has a significant negative relationship with both summer and fall sea surface temperature. So again, warmer fall, warmer summer, bad for surfclams. And those are just two of the many studies that have looked at warming impacts on surfclams. Now let's change gears for a moment to ocean acidification. So House et al. Found that for adult surfclams, scope for growth at a high or severe ocean acidification scenario, so a PCO2 of 2164 or a pH of 7.28, scope for growth at that scenario was significantly lower than at our two lower PCO2 levels or two higher pH levels. Now, Misek et al. Recently began addressing larval impacts with ocean acidification, and they found that larval surfclam growth at a high PCO2 or pH of 7.46 was significantly lower than growth at the two other carbon chemistry treatments. So the point here is severe ocean acidification negatively impacts adult and larval surfclam growth. So clearly, we know a lot, right? But there are still plenty of gaps out there. So that recruitment trend, I talked about how fall and summer sea surface temperature have a negative relationship with recruitment. Well, what's driving that relationship? Perhaps the larvae that will eventually turn into recruits that are spawned during those seasons aren't happy with the current temperature trends. We also don't know if ocean acidification and ocean warming interact with one another. We also don't know about other physiological responses. So for example, Misek et al. Found that surfclam larval growth decreased under severe OA, right? Well, why is that happening? Perhaps those larvae under the severe OA scenario are respiring more and using more energy for maintenance. Therefore, they have less energy left over for growth. Or maybe they're not eating as much because they're stressed out, a lower clearance rate, a lower feeding rate, which would explain the inhibited growth. And we also don't know how ocean acidification and ocean warming affect larval behavior. And the reason I'm interested in behavioral changes is because behavioral changes may lead to dispersal pattern changes. Let's talk about that for a moment with a brief oceanography lesson. So in our shelf waters in the Northeast coast, we typically have what's called a surface mixed layer. So this is the water layer typically above the thermocline that's highly influenced by wind. Because it's highly influenced by wind, faster currents, stronger flow. Now below the surface mixed layer, below the thermocline, we have a layer of water that's less influenced by the wind, if not virtually absent influence by the wind. That water layer, slower flow, slower currents. And so bivalve larvae, where do they come into play here? Well, bivalve larvae, they often swim. And when they swim, they often, but not always, swim up. And so the idea is that a bivalve larva that's swimming faster or swimming more frequently is going to be swimming up more and is therefore going to spend more time in this high flow surface mixed layer than a bivalve larva that is swimming slower and is swimming less and is therefore deeper in the water column and therefore experiencing flow below that surface mixed layer, slower flow. And so the idea then is that bivalve larvae that are swimming potentially and in the surface mixed layer may be advected longer distances and have different dispersal patterns and paths than larvae that are swimming slow and below the surface mixed layer and are experiencing less advection and perhaps different dispersal distances and paths. So that's the swimming behavior aspect of this. Another metric relevant to larval dispersal is what's referred to as PLD, or pelagic larval duration. You could think of that as a fancy way of saying how many days until the larvae settle. And the effect of PLD on dispersal may be a bit more intuitive. PLD, the idea is that a longer PLD means more time in the water column. More time in the water column, more distance traveled before settlement. Shorter PLD, less time in the water column. Less time in the water column, less distance traveled before settlement. So we care about these dispersal patterns because again, different conditions may lead to different dispersal distances, different dispersal paths, and different settlement locations where the larvae settle. Where we're going to have recruitment. Where we have recruitment, where we're going to have adults. Where we have adults, that's where the fishing has to happen. So now my two general hypotheses for this study. First one was that surfclam larval behavior and physiology were affected by observed ocean warming. And the second general hypothesis is that surfclam larval behavior and physiology will be affected by predicted warming and ocean acidification. And so to address these hypotheses, my colleagues and I set up a laboratory experiment. For our lab experiment, we chose 3 temperature treatments: 17 Celsius, 20 Celsius, and 23 degrees Celsius. These conditions represent hindcasted, present day, and forecasted conditions in the New York Bight during the spawning season of surfclams and surfclam habitat. And our 2 pH levels were 7.7 and 7.3, which represent present and forecasted conditions. I will admit 7.7 is a bit low for those waters. However, there are surveys out there that show that this value is found in shelf water in the summer, specifically closer to the inlets, where actually happens to be where a lot of the surfclams tend to hang out. And so the idea is, if we compare this scenario to this scenario, we can potentially explain that recruitment pattern. So again, negative relationship between summer temperature and recruitment. Well, maybe the larvae are stressed out by the recent warming in the past couple of decades. Maybe the larvae aren't happy with this 3-degree increase. And so if that's the outcome, that would explain why we have poor recruitment recently. And that would explain that relationship. Now we can also compare this scenario to predict what's going to happen in the future. However, these are just 2 of the general main comparisons to make based off of my specific hypotheses. However, of course, we can make all sorts of other comparisons between our different treatment combinations. And because we had a fully factorial design, we have 3 temperature treatments, 2 pH treatments. That leads us with 6 different treatment combinations and 5 replicate tanks per treatment, giving us 30 tanks we had to maintain. So for those who have not seen the OA lab at DEI, here's what it used to look like. It's at least double the size now inside, but this is a glimpse into the OA lab system. So you can see we have these 11-liter conical tanks. These are what hold the larvae. And these heat jackets around the tanks, that's what controls the temperature. And then most of the panels and wires and tubes you see up here, that's the apex system that controls our carbonate chemistry parameters. Okay, now the fun part, the responses that we measured. Boy, did we measure a lot. So let's start off with the basic ones, mortality and growth rate. Mortality we measured as the percent of dead larvae at the end of the experiment. So we didn't do like a Kaplan-Meier survival analysis. We just looked at mortality at the very, very end. Growth rate we measured as the increase in larval length through time. And we wound up just taking the length on the last day, length on the first day, dividing it by the total number of days to give us a rate in terms of microns per day. And it's that microns per day increase in length that we performed the analyses on. Clearance rate was measured on about 2-week-old larvae. The clearance rate is food consumption, right? So in this case, it's algae consumption. We measured how many algae cells an individual larva consumed over a few hours. Then we standardized it per hour, per biovolume of individual larva. Respiration rate, same idea. Slightly older larvae, 3-week-old larvae. But again, we were able to standardize the assay so that we're able to get this number of the amount of oxygen that's consumed per biovolume of individual larva per time, per unit time, in this case, 1 hour. Now, immune performance. Essentially, immune performance is mortality rate due to exposure to Vibrio bacteria. So what we did here is we kind of set up a separate assay, a separate trial with well plates. We put larvae in the well plates and exposed them to Vibrio bacteria. Of course, we had controls without the Vibrio bacteria. And the idea is if you set the experiment up that way, you can quantify mortality that's strictly due to bacteria exposure, theoretically speaking. So immune performance is just mortality due to bacteria exposure. Okay, and now biomineralization. We measured this at 3 different time points, on 16-day-old larvae, 23-day larvae, and 30-day-old larvae. We measured this on fixed dead larvae. So what we did was we took these fixed larvae, put them under a microscope, and exposed them to cross-polarized light. And this is the image you get. And what we actually measured was birefringence, which is the double refraction of light when exposed to cross-polarized light. So the idea here is that the brighter shell material is exhibiting more birefringence, suggesting higher biomineralization. And to actually quantify that, we simply put these images into image J and calculated the average grayscale value of an individual larva. So the idea is a higher grayscale value means more birefringence. More birefringence means higher biomineralization. Now on to our behavioral responses. So we measured swimming speed, real time, of course, on 4 different days: 5-day, 11-day, 16-day, and 23-day-old larvae. And we took video recordings that looked something like this. And we simply took those video recordings and input them into ImageJ. We used this cool plugin called WormTrack. Pretty simple plugin, and it just spits out the swimming speed in pixels per second of each individual particle, which in our case is a larva. Then you simply convert that to millimeters per second, and voila, you have your swimming speed. Percent swimming, same idea, same videos. We just calculated the percent of larvae that were swimming before the end of the video. Pretty simple. And that too was measured on the same days as swimming speed. So we have some younger larvae, middle-aged larvae, older larvae there. Now, pelagic larval duration, or PLD. So what I did here was, starting on day 20, every day after that, I would take about 50 larvae from each tank. I would then put them under the dissecting scope, and I would calculate the percent of larvae that were exhibiting what I call foot action. And this is what I mean by foot action. And if the video wants to load, it's there. You can see the feet of these 2 larvae are looking for substrate to settle on. And so because I did this every day from 20 days on, I was able to predict, or even sometimes measure, the day at which 50% of the larvae were exhibiting this foot action. And it's this date at which 50% of the larvae are seemingly ready to settle that I'm calling the pelagic larval duration. Okay, now on to the results. First, let's keep it basic and talk about mortality. So our 2-way ANOVA for mortality showed no significant pH effect and no significant temperature effect. And if you look at the data, that's not too surprising considering the high amount of variability here. We have some tanks that have near 100% mortality and some as low as 50. Nevertheless, data all over the place, too much variation, no significant effects there. However, immune performance, which again is mortality due to bacteria exposure, did show a significant pH effect. No temperature effect, but a significant pH effect. And in fact, we see that mortality due to bacteria exposure was higher for larvae in the acidified treatments than in the non-acidified treatments. Growth rate. So what I have plotted here is length through time, right? But what we actually conducted the ANOVA on was just the final change in length through time. So in other words, took this number, subtracted this number from it, and then divided it by the number of days, and that rate is what we performed the ANOVA on. And so our ANOVA showed us that we did have a significant interaction between temperature and pH. And so then Tukey's multiple comparison test showed us that growth at 20 degrees Celsius and a pH of 7.7 was significantly higher than all other treatments. And we saw no difference between any of those other treatments. So the idea here is warming and no acidification, high growth. All other treatments had lower growth. Okay, now PLD. So again, what I did here is I used those measurements for the foot action to predict the date at which 50% of the larvae were going to settle. And for that time series, for some preliminary analyses, I just used GAMs to try to make a model to get to that prediction. What we can see, let's go into the 23 Celsius and 7.7 treatment. So that treatment led to the shortest PLD, 26 days until half the larvae were settling. It's not too surprising when you consider that this treatment also exhibited the highest growth rate. Now let's jump to the acidified version of that treatment, so 23 degrees Celsius and lower pH. We have a much longer PLD of 31 days. Now let's look at the two 20-degree treatments. 20 degrees and acidified, PLD of 29 days, and 20 degrees and non-acidified, slightly longer PLD, 32 days. And then our two coldest treatments, both non-acidified and acidified, showed PLDs of 34 days. Now into percent swimming. Because we measured percent swimming in multiple days, we have a three-way ANOVA with time being our third factor. So for percent swimming, we did see a significant time impact. Not too surprisingly, the amount of larvae that were swimming when they were 5 days old was significantly lower than all the other days. However, we did not see a significant temperature or pH effect for percent swimming. For swimming speed, we again saw a significant time effect where younger larvae were swimming slower than older larvae. We again did not see a significant pH effect, but we did see a significant temperature effect. More specifically, we saw that swimming speed at 23 degrees Celsius was significantly lower than at both our 20-degree treatments and 17-degree treatments. Biomineralization, and this also occurred multiple time points. So we first started off with the three-way ANOVA. However, we saw significant interaction between time and pH, as well as time and temperature. So then we simply just performed a two-way ANOVA on the individual time points after, of course, a Bonferroni correction. However, the ANOVA output I'm talking about right now is strictly the two-way ANOVA output for our day 30 larvae. And for our day 30 larvae, we saw both a significant pH and temperature effect. We saw that the biomineralization values, that grayscale value, right, that tells us about birefringence, was significantly lower at a pH of 7.3 than at a pH of 7.7. We also saw a significant temperature impact where biomineralization at 17 degrees Celsius was lower than our two other temperature treatments. So again, 17, lower biomineralization than 20 and 23 degrees Celsius. Now, respiration rate. Respiration rate, again, was on about 3-week-old larvae. We did not see a significant temperature effect on respiration rate, but we did see that respiration rate was significantly higher in the acidified treatments than in the non-acidified treatments. Clearance rate, slightly different story. So we didn't see a significant pH effect for clearance rate, but we did see that larvae exposed to the coldest treatment, the 17-degree treatments, did clear less algae, did consume less algae than larvae at our 20 and 23 treatments, 20 and 23. So in addition to all those ANOVAs and the one preliminary GAM analysis, we also conducted principal component analysis. The reason we did this was to kind of get a holistic idea of the energy budget profile, to see how responses that may be directly related to energy acquisition and expenditure may be related to one another. So the 5 responses we included in our PCA were biomineralization, growth rate, clearance rate, respiration rate, and swimming speed. So the 2 principal components explained about 65% of the variation. Not the best, but also not so horrible. And our clustering analysis yielded 2 clusters. So we had this one cluster over here containing exclusively the high pH and high temperature treatments. And then all the other data points were grouped into the second cluster. And the PCA only wanted 2 clusters. But we can see if we want to kind of divide this into subclusters just visually, we can see there is, the acidified treatment data points hanging out up here and the non-acidified treatment data points hanging down here, with this one exception. What I think the most interesting takeaway message here is how the different responses are related to one another. So if we look at the vectors for each of our responses, we can see growth rate and biomineralization. Those 2 vectors are kind of going in the same direction. That tells us that these 2 responses may be positively correlated to one another. In other words, when we had higher growth, perhaps we had higher biomineralization. Now we can see that clearance rate forms about a 90-degree angle with both growth rate and biomineralization. This means there is likely no correlation between clearance rate and these two responses, which is a bit odd considering clearance rate should be related to growth rate. And then if we look at respiration rate and swimming speed, these two responses form somewhat of a 180-degree angle with growth rate and biomineralization. That tells us that these two responses may be negatively correlated with growth rate and biomineralization. In other words, when respiration was higher and swimming speed was higher, that meant growth rate and/or biomineralization was perhaps lower. Okay, now into the discussion. I first want to talk about the larval transport implications from the specific results from this study. So present day scenario, this would be the 20-degree treatment and pH of 7.7. That treatment yielded relatively high swimming speeds, right? So those larvae are going to spend some time in that surface mixed layer, presumably, and therefore experience a fair amount of high advection. That treatment also had a relatively long PLD of 32 days. So when you combine the high swimming speed and the high PLD, we may have a relatively long dispersal distance, perhaps 100 kilometers. And this number comes from a biophysical modeling study that simulated transport of surfclam larvae. And I'll show you some results, some figures from that study later on in the discussion. Just wanted to let you know, this number isn't completely fictional. And then let's fast forward to a warming and acidification scenario. Here, we didn't see much of a drop in PLD, only dropped by 1 day, but these larvae swam significantly slower. So these larvae, presumably, theoretically, may be spending less time in the surface mixed layer, more time deeper in the water column where there's a slower flow. Because of that, these larvae may have a bit of a shorter dispersal distance, perhaps closer to 80 kilometers as opposed to 100 kilometers. And then our warming scenario only, not only did we have slow larvae, and therefore larvae, again, theoretically below the thermocline in that slow flow zone, but we also had larvae with a much lower PLD. When you combine the low PLD with the low swimming speed, you may have an even shorter dispersal distance, perhaps closer to 60 kilometers as opposed to 80 or 100 kilometers. Enough about larval transport for a moment. Now let's switch gears to the energy budget and some relationships and perhaps some mechanisms going on there. So we saw that there was high growth under warming only relative to all other treatments, including the acidification and warming treatment. So why is this the case? Well, we did see that there was higher respiration under ocean acidification. So it's possible that when the larvae were exposed to these low pH waters, they were using so much energy for maintenance that they had less energy left over for growth. The results of the study support that hypothesis. Now, we also saw that larvae under the warming scenario had a higher clearance rate, so they had higher energy acquisition. That makes sense, but it doesn't really explain then why growth was higher under warming only as opposed to warming and acidification if we didn't see an acidification impact on clearance rate. We also saw a lower swimming speed under ocean warming. So it's possible, and the PCA supports this, that swimming speed is part of the energy budget profile. And when there was lower swimming speed in those high temperature treatments, specifically the warming only, there was more energy left over for growth. So let's talk about the swimming speed results for a bit. So we saw lower swimming speed under high temperatures. To be frank, this is weird. This is contradictory to both other results in this study and other studies. And what do I mean by that? Well, we saw higher temperature increased growth, which suggests that higher temperature increased metabolic activity. Well, if metabolic activity is higher, the larvae should be swimming more. That's typically what's found. And other studies that have looked at temperature effects on invertebrate larvae, including bivalve larvae, typically find that increased temperature increases swimming speed unless you eclipse that really high temperature threshold by which the larvae are stressed out. But we presumably didn't reach that threshold because the larvae had high growth at 23 degrees Celsius. So what's going on here? A couple of potential reasons. So while the ocean warming scenario in this study increased metabolic activity, perhaps it disrupted the sensory functions of the larvae. This may be important because cilia control swimming behavior. Cilia directly tied to sensory functions. So that explanation makes some sense, but I don't know if I buy it that much, because sensory functions and bivalves are typically strongly influenced by acid-base regulation. So if that was the case, we would also likely see an acidification effect on swimming behavior, which we didn't really see. What may make more sense is taking kind of a big picture environmental perspective and thinking about cues and perhaps food. So let's say you're a bivalve larva. You're in warm water. You say, okay, it's warm around me. It's warm, that means I'm probably close to the surface, or at least above the thermocline. If I'm above the thermocline and close to the surface, that means I'm in an area with presumably high phytoplankton. If there's more food around me, I don't got to swim as much to get to that food. But now, if you're a larva and you feel that it's cold around you, you may say, oh, okay, it's cold around me. That means I'm deeper in the water column, below the thermocline, below the surface mixed layer, where there's less food. Perhaps now I have to swim faster to get to those areas that have more food availability, which may be related to temperature. What are our general conclusions here? Well, my first hypothesis of trying to explain that New York bite recruitment pattern or relationship, again, warm summer, warm fall, poor recruitment. I thought maybe that's because larvae aren't happy. It's not the case. We saw no significant difference between 17 Celsius and 20 Celsius in terms of growth and mortality. So there's something else going on with the recruitment story. For ocean warming only, we saw that was good for larvae, more specifically ocean warming only at 23 degrees Celsius increased growth rate. So it seems that the larvae like it above 20 degrees Celsius, whereas the adults don't like it above 20 degrees Celsius. Not too surprising when you consider the size difference and the amount of energy that needs to go into maintenance for the two different life stages. For ocean acidification only, we saw that this may be bad for larvae in that it led to higher immune mortality or mortality due to bacteria exposure, led to higher respiration rates, so higher maintenance demands, and a lower biomineralization index. And then for the warming and OA projections combined, relative to ocean warming only, we saw that this was bad for larvae in terms of growth rate. In other words, growth at OA and warming was lower than warming only. And going back for a brief moment, conclusions related to dispersal patterns. So under warming and acidification together, there may be a moderate chance for shorter dispersal distances, as this scenario led to lower swimming speeds. And then for the ocean warming only projection, we may have a higher chance for shorter dispersal distances, as this scenario led to both lower swimming speeds and a shorter PLD. So next steps. I talked about a study that simulated transport of larval surfclams. Well, these are two figures from that study. We kind of see this down coast movement of larvae. Georges Bank supplies some larvae for itself, as well as larvae for southern New England. So New England supplies larvae for Long Island for itself, New Jersey, and so on and so forth. So that shows us the general patterns of dispersal. Now, in terms of distances, we can look at the box plots for these different regions, and we can see that the median dispersal distance for our different regions tended to hover around 100 kilometers. So that's where that number came from earlier. Well, I want to see if we put in climate change induced changes in behavior and PLD, do these patterns change? Do these dispersal distances change? And if so, by how much? And so the conditions I focused on in this experiment, specifically the temperatures, were really focused on the New York Bight region. That's only one area that surfclam larvae are found in. They're found all the way down to Cape Hatteras and up into the Gulf of Maine. I want to try to quantify the full thermal niche of surfclam larvae and see how development changes through time and space due to the wide range of temperatures they may experience through time and space. I also am interested in the role of food availability. Now, I was kind of alluding to that with trying to explain some of our behavioral results. But more specifically, what I want to see is just like temperature, there's natural variation in food availability. So amount of food, in Georges Bank versus Gulf of Maine versus shelf waters in the Mid-Atlantic Bight, does that variability impact surfclam larval development? And that's part of what I'm doing this summer. And so I have a little teaser for you. here we have growth rate of surfclam larvae, microns per day. And then on the x-axis, we have total chlorophyll concentration. And I put relative approximate labels for some of the different regions that surfclam larvae are found in. We can see a significant relationship between surfclam larval growth and total chlorophyll concentration. More specifically, we can see that growth was, not too surprisingly, higher at higher chlorophyll concentrations than at lower chlorophyll concentrations. And so before I take questions, I want to try to do some recruitment, pun intended, for next week's speaker, Alex. She is my fellow here at DEI from Rutgers, and she'll be talking about her work regarding relationships between ecosystem services and aquaculture gear. It should be some cool stuff. And as you can see here, she'll have cooler pictures and videos to show than I did. So hope to see you all next week for that. But now I will gladly take any questions. Looks like there's maybe one in the chat. Oh, that's just.
Does anyone have any questions for Ray? Ray, that was a really comprehensive overview of different impacts to surfclam larvae. Thank you.
Oh, I think I see a hand here. I don't know.
Hi, Ray.
Oh, hi, Daphne.
I have a couple of questions for you, if nobody else has questions to start off. First of all, really cool work, and it's good to see you continuing these experiments. My first question is about your bifringence metric. How, because you get a bit of a curious result out of that, in that your biomineralization goes down as the larvae get bigger, which is the opposite of what I'd expect. Did you validate the cross-polarization metric you were using, the bifringence metric, with actual shell biomineralization in any way?
So
were you able to do that?
By actual shell biomineralization, you mean like SEM or something?
Yeah, or anything. Yeah, was there any way for you to actually test your bifringence response?
So that's all that we did. You know, I showed you everything. The way I look at the bifringence response there is, you know, okay, lower bifringence, lower biomineralization, and then potentially, like, why is that the case? And there's a few reasons. Perhaps it means, you know, actually thinner shell material. Maybe it means more porous shell material. Maybe it means different structure, crystalline structure. And so I think, you know, those would almost be the way I think about it. Those may be the mechanisms for the bifringence, whereas the bifringence is just kind of a general assessment of, you know, how healthy and happy the shell is. The way I interpret the trend through time, I'll go to that figure now, is, you know, I just think the larvae were not particularly happy in our experiment. You know, we had very high mortality. And so I think the larvae tried to hold on as long as they could, but then eventually they just said, no, okay, I'm done trying to produce shell material because I'm stressed out from this lab experiment. And that's why the index drops through time.
Okay. Moving to another question then about your swimming response. You get lower swimming speeds under higher temperature, and you gave a couple of really interesting explanations about that. And I think particularly the one about stratification is, you know, maybe is an interesting one about cues of where you are in a stratified water column. But Reynolds number would change with temperature. Did you take any of did you account for possible changes in Reynolds number across these temperatures?
I didn't, but if my understanding and recollection of those aspects of physics is correct, if there was an effective Reynolds number, shouldn't we, I think I should have seen the opposite, right?
Reynolds number would go up, right, with increasing temperature,
Let's see, I know.
which would mean that it would become a more, it doesn't, no, I'm going backwards, I think. Reynolds number should go down with increasing temperature. I have to remind myself which way that goes though, Ray. Yeah.
I remember thinking about that, and the end point of my thought process was that would explain, you know, that would explain if it was the opposite, if temperature increased swimming speed. So I think viscosity decreases at higher temperatures.
Right.
It's easier to swim at higher.
So it would, yeah, it would have gone the other way. Okay. And then one last question, if there's nobody else with their hand up yet. On your experiments that you're doing this summer, your chlorophyll, there seems to be a gap a bit in your chlorophyll numbers. You've got some low numbers that are close together, and then you've got that coastal Gulf of Maine. Can you explain what's going on there in your treatments and how that's being set up?
Yeah, so what you're looking on at the x-axis here is total chlorophyll concentration. However, as some of us may know, those total chlorophyll concentrations that come from, you know, satellite data include things from picophytoplankton to, you know, huge dinoflagellates. Bivalve larvae really only feed on a fraction of that total chlorophyll. You know, we can say that they feed on the 2 to 20 micron stuff, the nanophytoplankton. And so there's a recent study published last year that looked at the relative contribution of those 2 to 20 micron phytoplankton to the total chlorophyll concentrations that are captured by the satellite data. And essentially, they found that once total chlorophyll, essentially, these numbers could be really anywhere here. Once you hit about 4 total micrograms of chlorophyll per liter, all of that's not going to change the biologically available phytoplankton for surfclam larvae. Once I get my hands on the exact model that paper used, I can, you know, pinpoint this, but these dots can, you know, probably can be anywhere close to over here too. Does that make sense?
Okay. Yep. Thanks.
Brian.
Sorry.
Brian has his hands up.
Right, so Ray, this is Brian. I agree with Daphne with a really nice job of presenting this data. And you somewhat hinted that your results from your current study are helping you or informing a little bit more about what your results mean from the 2021 studies that you've presented here. I'm just wondering, could you say a little bit about what your current studies doing to inform the results that you presented here?
Sure. One of the more obvious things I've seen so far relative to what I found last summer was we found that 23 degrees was a good temperature for a surfclam larvae. They like that. And so then after the last summer, I was thinking, okay, well, what's that tipping point? You know, what temperature did they become unhappy? And it seems right after 23 degrees is that tipping point. You know, we had a whole range of temperature treatments, and I think our 24 and 26 degree treatments had significantly lower growth. The other, maybe perhaps a better way to explain it, is I saw a quadratic relationship between growth and temperature, with the apex of that relationship being at 23 degrees Celsius. So that's giving me a good idea of that full thermal niche that I was looking at.
Super, thank you.
And I'll ask one, Ray, or have a suggestion or comment, I guess. At the end with your conclusions, you noted that it didn't appear that the ocean warming effects on larvae were the explanation for the declining recruitment. And so I guess I will just put in, and I think I made this comment last year to you too, about maybe an indirect result of warming in terms of like predation and how that factors in. And you've looked at a lot of different things with your model. So there's a lot going on, but that might be some sort of explanation to also look into.
Yeah, no, I do think predation is part of the picture there. Unfortunately, it's hard to do, the types of field experiments we do up here in the intertidal with Maya and green crabs, doing those types of things in 50 meters of shelf water in the middle of the New York Bight. That'd be very difficult to try to get at the role of predation there. That being said, predation is thought to be a big factor in surfclam recruitment. And, there may be data sets out there that show abundances of some of the surfclam predators as time series. There's none that I could find for the New York State DEC survey data, but maybe at the federal level, there's some data sets out there that could help inform that potential relationship. But I do think it's part of the picture, yeah.
Great. So does anyone else, are there any remaining questions for Ray? Sort of a lot of information being presented at once, but it's really great to sort of get a comprehensive look at these factors that are affecting larvae. So if there's not, Ray, thank you so much for such a great presentation. This was awesome. Ray is also going to be presenting another seminar at the tail end of our series on August 16th, where he's going to talk about his results from this year. Oh, sorry. No, that's not true. You're going to be talking about your model.
A meta-analysis that I'm working on. Yeah.
Okay.
So something very different than anything I've kind of hinted at today or even last summer.
Okay. Yep. And so please think about joining us next Tuesday. As Ray pointed out, our second DEI fellow, Alexandria Ambrose, is going to be presenting her work on understanding how vertical muscle long lines can create habitat for organisms. And we'll be meeting again each Tuesday until August 16th. And I put the schedule of the upcoming seminars in the chat for people. And I'll also be sending out notices. So I'd like to thank everyone for coming. And thank you again. If you want to get a hold of Ray, I can also add his email to, I can also add his email to the chat. So thank you all.